Lopez Martinez, Christoph Gumosowski, and Anne de Blick. Please take a chair. So, please get comfortable, Fred, for accepting this move. So the subject of our little panel is structure-activity relationship. And before we come to any question, I would like to ask the panelists to introduce themselves, their company, and what they or their company has to do with structure-activity relationships.
Good afternoon. My name is Manuel Lopez. I'm originally from Barcelona. And I've been working for Amirai for more than 22 years in computational chemistry, together with a chemist, the BK department, and metabolism. And now, 3 years later, I was moved to a French company. It's called AB Science. It's a small pharmaceutical company. It's dedicated to work in oncology and kinesis. My experience is, I've been working as an organic chemist, medicinal chemistry, for 3 or 4 years. And then the rest, I moved to computational chemistry, and I work in that field.
Thank you very much. My name is Thorsten Hoffmann. I'm the Chief Scientific Officer of Silence Therapeutics. We are an RNAi-based company. I'm CSO there since 2017. Before that, I was 2 years Chief Scientific Officer of Proteros Biostructures. Then I was, before that, 2 years Chief Scientific Officer of Zealand Pharma in Copenhagen, Denmark. And before that, I was 16 years at Roche. The past 8 years, I was head of medicinal chemistry at the headquarters. What have we to do with SIR? So I counted the projects I've been involved in. These are 96 projects. And we worked on small molecules, we worked on peptide medicines, we worked on siRNAs and their conjugates. So I think that there has been always SAR involved, and it's perhaps also worthwhile to mention in my introduction, I'm inventor of a marketed medicine. Out of those 96 projects, 1 made it to the market, so that's so much about statistics. Mm-hmm. And I also calculated or looked it up, so 60 out of these 96 projects came to the lead series identified stage, and a bit more than 25 molecules have been filed as INDs.
It's nice to come after you.
So we talked about the experts. Now
Exactly.
I'm looking forward to hear your perspective.
So yes, my name is Anne de Blick. So I'm a senior scientist, medicinal chemist. I worked first for 5 years and a half at a biotech company Galapagos, based in Belgium, France, and the Netherlands. And since last year, I'm working at a small biotech company, Comfo Therapeutics, which was funded in 2015. So here our focus is actually on finding new drugs for GPCR. So in the past at Galapagos, I worked, when it comes to structure-activity relationships, on a kinase project. I also worked on an antibacterial project with a macrocycle. I worked also on an antiviral project, but there the target was unknown, so it was a phenotypic screen. So, but still you learn from your data and how you try to improve your molecule. And then now, of course, at Comfo it's GPCRs, but you all know we are looking at agonists, so compounds that activate the GPCR protein, which is not easy to get them in a stable form as well. So with our technology, we try to stabilize them, and as such then also allow fragment-based screening, and of course when we will be able to generate crystal structures, also go to structure-based drug design.
Thank you.
Hello, my name is Krzysztof Gumowski. I'm really honored to be here in this panel expert because I just graduated my PhD half a year ago. And around a year ago I joined Captor Therapeutics, which is a small biotech company based in Wrocław, Poland, which is working on induced protein degradation, and we are aiming at targeting difficult to drug proteome by the development of bifunctional degraders. And I think that the structure to function interaction is very well pronounced in the field of proteins, so I can give a lot of examples in that field.
Thank you very much for this round of introduction. And Christoph, don't be afraid. We want to have somebody here who is not stained by experience, who has a fresh look on sector activity relationship. So this will be your role. So relax, you are fine here. So before I open it up for the audience to ask questions, and I'm building on you that you are asking indeed questions, I want to kick it off by asking the panelists what in their experience the biggest mistake in structure activity relationship work is. So Thorsten, you have been involved in so many projects, and we know as somebody in pharmaceutical company, you have seen that there were some predictions, and at the end it turned out not to be true, and maybe you have some lessons learned you can share here with the audience.
Yeah, perhaps two answers to this. So one is the so-called activity cliff. It's really hard to predict and really hard to understand. So you're navigating, and here I'm talking now more about small molecules, you're navigating in molecular space, and then you seem to start and understand the SAR, and then you're making a minor change with enormous consequences, and you're losing all the activity. So that's in terms of, it's not really a mistake, but I always was keen to understand what causes activity cliffs, how can I predict them, and the other way around. If I'm very close to an activity cliff uphill, I'd like to understand what is required to go this extra mile through a minor change. That's the first answer, and the second one comes more what we discussed earlier. So where have we had a real failure? We worked on metabotropic glutamate receptors 15 years ago and brought a molecule into clinical development at a very low dose where we thought this is the starting dose. It was extremely neuroactive, it was psychoactive. The people reported they couldn't sleep. The people reported that they had suicidal thoughts. One person has been killing the Pope. So this was really a psychoactive molecule. And we couldn't predict this. And then we went back trying to understand what's wrong with this molecule, because it's only nanomolar. Then we found out we didn't look at the kinetics. And when we looked at the kinetics, we saw it's binding the target for more than 24 hours and super tightly. So that was a failure.
Okay. Anybody else want to add here? What were failures they have seen, experienced in such activity relationship work?
I think it's a bit related to that also, the activity cliffs, because in the antibacterial program with the macrocycle, it was difficult and challenging chemistry to start, but there also we had a crystal structure, but it was a tripartite crystal structure because it was a DNA polymerase, it was the molecule and the DNA, so seeing where it interacts and quite complex, but then also there, based on what we saw, we said, okay, there are key interactions there, and there. If we want to be active against more bacterial species, it's conserved because that part is the same if we go for that. But then when you try all those efforts and it doesn't result in it, not even speaking about that gram-negative are more difficult than gram-positive, but also just looking along the gram-positive species, even if you have a structure, it doesn't always help you to get the result what you want, and it makes it really tricky to understand, okay, what do I, it's all the information that I have, and then you're basically stuck. So.
Okay, thank you. Manuel, do you want to add something here?
Yes, I have one example with one particular kinase that, first of all, we get the compound. The compound was initially very active. And then we decided to run the crystallography of the compound, and we start doing modifications. And all the modifications that we ran were inactive. And this is something that we don't understand. Till we observe, after a collection of complexes, the role of the water. There was one water molecule inside an hydrophobic cliff that was conserved in all the crystals. And then, when we just only change, we introduce an acceptor in this particular place, we get another time the activity. And that's when we get finally the hit on the lid. But the role of the water is something very important, and we don't have enough knowledge of it.
Yeah. And still not computer means to calculate the water shell, right?
No. There are some softwares that can estimate using free energy perturbations what the waters can be or not. But I'm not really confident with the softwares because using free perturbation analysis, you can analyze small changes at a time, okay? And then, the water is something that is, you can displace it, and in one compound you can have one water molecule that is active, and in another complex you have solvation, free molecules, solvation, dissolution. Then it's not one change at a time. Mm-hmm. And this kind of techniques can give you an approach, but obviously if you want to know which is the real water, you need to crystallize each time the compound to see exactly what the water are.
To add there something from your background? Yeah.
So what I would say might be a cliche, but I think that one of the most important things that people sometimes forget when working in drug discovery is that, for example, when you obtain a structure of a protein with a compound, it's just a snapshot of a protein. And we forget that the proteins are very flexible and they can really change. And also one of the pitfalls is that sometimes if we cannot crystallize a protein, we will cut the flexible parts and then we work only on a domain. So this is, I think that these are the challenges in this field.
Is it working? Yeah, and to add my personal experience from my youth, when I discuss crystal structures with people from computer modeling, they always look very nicely and very neat, and there is not much space available. Then you add something, and the prediction was, okay, this will not fit, but it fit. And then they said, okay, there was an induced fit. This is difficult to anticipate. Have you had similar experience?
Yes, indeed. They say, and if you go for this and this interaction, or they say in their model, yeah, but if you put a bigger halogen there, it will not work, and then all of a sudden you make it, and your activity is even better. Yeah, but it twists. It's then different. You have freedom. It can go in different ways in the binding pockets, and if you put that, it goes around. So.
Okay.
For this kind of examples, for example, I prefer the ensemble doctrine, is to use the picture of a lot of different complexes that can take into account a little bit the movement of the residues, and then you can have a better picture of the interactions. But that works relatively well when your active site is very well defined, for example in kinesis. But I have experience in GPCR, and you have the famous loop. Mm-hmm. Okay, and that loop is moving all the time in everywhere. And then it's impossible to make any prediction. It's very difficult, or you can have an approach, but it's very difficult. Mm-hmm. Then it depends. You have to analyze very well which kind of active site you have, if it's very exposed to the solvent, not exposed to the solvent. And if for each particular case, you have to apply a different approach.
Mm-hmm. Okay, thank you. Is there any questions from the audience? If not, I have a second one, but then I'm running out of questions, so I need your help, guys. I buy you 10 minutes, but then you are on. Yeah, thank you.
When Torsten was talking about this activity cliff, it puts me in mind of some problems that we've had, which could impact upon SAR, although they're not quite the same. And this is the process of or a phenomenon called hormesis. Now, I don't know whether you know hormesis, but it's basically the phenomenon which we've encountered quite regularly, even though we've been working only with a small number of molecules. It seems to happen particularly with peptide drugs, where you get an activity which is okay in the animal, and you think that you're going to improve it by increasing the amount of drug that you give to the animal, and it goes down actually, and then you reduce the concentration and the activity goes up. And I don't know how many. When I spoke to some people, they say, "Oh yes, peptides do that." I don't know how much experience there is. I'd be very interested in the experience of other people here, because this is something which some of my colleagues have said, well, if that's the way that it behaves, you're going to get a problem with the regulatory authorities. Yeah. Now, how can how can we have you have a problem with the regulatory authorities. You have a problem with some people in the large pharma as well. But it's a common phenomenon. How can one adapt our thinking and adapt other people's thinking to make a success of these drugs which behave in that way, which is actually quite a common phenomenon?
Yeah. So I can give you an immediate answer to this. When I was CSO at Zealand Pharma, we worked on can I mention the target? Probably not. So we worked on agonists, and we found exactly this. If we went up in dose, the effect was going away. So the pharmacologically effect in vivo was going away. And then what we started to do is we dosed every second and later every third day. And then the activity came back. And I think the explanation we had at that time was the pharmacology of the receptor. When you overstimulate with a higher dose and daily, then that leads to receptor internalization. The receptor is gone. No effect can be exerted on the cell, on the cellular level. No signaling occurs. But if you allow for receptor recycling into the membrane after you've stimulated this, then we had a huge pharmacological effect. It was actually weight loss in that case. And the animals, they lost a lot of weight when we dosed them only every second or third day. So that was our explanation to what you also described on peptide agonists, probably.
Yeah, thank you.
Yeah,
Further questions?
just commenting on that, I think that, yeah, I mean, there are probably many explanations, but what I was thinking about was autoreceptors, that if you that you actually get a different pharmacology from if you have a low dose, you just touch the outside, and there you have the autoreceptors. But when doses get higher, then you touch the inside of the synaptic cleft. That could be, but I mean, yeah, then desensitization and that I fully agree.
Or on specificity that you at a certain dose range hit another target which counteract your target. That's also a quite common phenomenon here. Another point, I think, when we talk about such activity relationship, we need to talk about on which level of activity such activity relationships should be built up. You know, you can work out your such activity relationship on the isolated enzyme, which is in a way very artificial surrounding environment, and on the other side you can go probably in full cells, which makes it more complicated because a lot of factors get into your accreditation there. So what do you think, where's the sweet spot? How complicated assay needs to be meaningful for advancing the right molecule to the next level? What do you think?
I'm thinking about it.
So Thorsten, you are the most experienced one here in our panel. Maybe you can start.
So you're saying I'm old.
So just an information to the audience. Thorsten and myself, we made together a postdoc in San Diego. We are roughly the same age.
We are. We're actually great times.
Yeah.
So the sweet spot is probably the cellular system, although I'm also a fan of phenotypic screens where you're really resembling already a kind of disease state. So to give an example, if we had a pancreatic beta cell which fails to produce insulin, so that's kind of resembling what's really happening in the disease. Because when a diabetic is diagnosed, 75% of the cell function is gone. So if we had a pancreatic beta cell as a cellular screening system, diseased cell, and we are screening for molecules that are restoring insulin secretion. So that would be for me the kind of ideal, and I asked the people at Roche a lot about this. We never had it, but that would be the sweet spot.
Yeah, but beta cells are not the most easiest cell to work with, right? Okay, but we got this. And you worked on a phenotypic screening, you said in your introduction.
Yes, so that was for an antiviral project, so it was with liver cells, but there also we then looked at different types of liver cells. We started with the HepG2 because they're not metabolically active to look at your biomarkers, but then of course you don't really have the information what happens to your compound, so then you go to a HEPA-RG system which has more metabolic activity, and then finally to human hepatocytes, which are also not easy then to work with, and you depend on the fresh cells from a patient to immediately use. But I think indeed that a cell system still gives you the most information of it's the same for kinases. You can look at the kinase itself, or you look at the whole blood assay for your selectivity difference that you get, and it can sometimes be completely different from if you have a great selectivity in your isolated